Blood groups. Blood groups were discovered in 1900 by the Karl Landsteiners, and he discovered there are four types of the blood group. That is the blood group A, B, AB, and the O. These blood groups are divided according to the presence of antibodies and the antigens in the surface of the RBCs and the plasma. The blood group O have no surface A and B antigen, and the anti-B and anti-A antibodies are present in the plasma. In the blood group B, they have surface antigen B and they have the antibody A. Blood group A, surface antigen is present A and the antibody which is present in the plasma is the anti-B. And in group AB, they have surface antigen of the A and the B and they not have any anti in the plasma. Now the ABO blood grouping, there are four types of the blood group, right? A, B, AB, the O. The blood group O is called universal donor because it has no antigen on the surface. The blood group AB is called the universal receiver because it has no antibodies in the plasma. Now, there are another factor which are present and which differentiate the blood group is called the Rh factor. This is also an antigen, and those who have the Rh factor, they are called the Rh positive, and those who not have any Rh factor, they are called the Rh negative. Now, something about the anti-sera. When you start doing the practical of the blood grouping, you have the anti-sera. There are three type of the anti-sera, and you can see in this picture this anti-A, anti-B, and the anti-D. And these anti-sera, they have different type of the colors. The anti-A, blue color, the anti-B, yellow color, and the anti-D, the B color. So when we start the practical, just prick your finger after all antiseptic measure. Take a tie, white tie or slide, and mark on that slide A at the corner of the slide, mark A, B, and the D with the help of the marker. Then put a drop of the blood in front of the mark A, one drop in front of the mark B, and the one drop of the blood in front of the mark D. Now add a drop of the anti-sera A in the blood drop of mark A, anti-sera B, the blood drop of the mark B, and the anti-D sera in the blood drop of the mark D. Then mix the anti-sera with the drop of the blood slowly with the separate mixture. You can take toothpicks or anything and mix it separately. Take the separate toothpicks and wait for the agglutination to occur. Now you see this picture. In A positive blood group, if you have the A positive blood group, there is agglutination or coagulation occur in the drop of the blood of the anti-A and the D. It means A and D both are positive, and so you call A positive. And when you have the blood group B, you see the agglutination is present in the blood drop of the B and the anti-D. So you have the B positive. Then if you have agglutination in all these three drops, means A, B and the D, it means that your blood group is AB positive. And if you have not have any agglutination, it means that there is your blood group is O negative. And same if you have the coagulation in the anti-sera A and no coagulation in the anti-D, it means you have A negative. And if you have the coagulation in the B and there is no coagulation in the anti-D, it means you have the blood group of B negative. And in case of the O positive, there is no agglutination in A and B and there is agglutination in the anti-D. So the blood group is O positive. You see in this case B positive and difference between the B positive and the B negative.